Hi guys, and welcome to another video in the HSC chemistry series on production of materials. In this particular video we're going to be focusing a little bit on the fermentation of ethanol. Now this is an experiment that you should be carrying out in class anyway, so just a little bit of background and a comparison between the laboratory process and the industrial process are going to be the subjects of this particular video. So the reason we're talking about this, really, the reason that we're linking this into our study of ethanol is because of the fact that ethanol is a renewable resource. And we've contrasted the fact that fossil fuels are a non-renewable resource. They take a long time to form and a very short period of time for us to use them. So therefore, we are replenishing them at a much slower rate than they can actually be produced. This is not the case for ethanol, and certainly ethanol that we can obtain from the fermentation of plant material. So it allows us an alternative for the production of ethanol, either for use as a fuel substitute or also for conversion into ethene and therefore the start of development of different polymers. So what is fermentation? Well, it's basically a biochemical process that some organisms use to convert glucose into energy. It's a form of anaerobic respiration. I shall write that for you, anaerobic respiration, because it's a generator of energy, and it has several important conditions associated with it. The first of those is the presence of a microorganism that is able to carry out this process, and in our case that's going to be yeast, which is a particular type of fungus. It also requires an aqueous solution, so we need some simple sugars like glucose or sucrose for us to grow the yeast. We also need anaerobic conditions, which is no or at least very low oxygen. And we also need warm temperatures, around 30 to 35. So typically what we would do is we would put that into in a laboratory, into an incubator, or you could use a heat mat or something like that, just to try and maintain that temperature at an optimum for the functioning of the yeast. Now industrially it's slightly different. Obviously it's much larger scale. The production usually comes from biomass, large material or matter that's locked up in biological organisms that usually come from things like sugarcane or corn. These can be grown fairly simply. They're fairly simple plants in terms of their growth. They can be grown over large areas in large quantities, large biomass, and hence they can be used as the start of the fermentation process. So the first thing we have to do is to crush the corn or sugarcane into a rough paste. Waste molasses is what we get from the sugarcane. So that's just a sort of bit of a sticky mess. The cellulose and the starch that are present in those plants, that sort of paste or molasses, is then converted into simpler sugars. So these are both complex sugars, the cellulose and the starch, and we need them to be simpler ones, such as glucose, because that's easier for the yeast to actually work on. So we do this via either enzymes, which can directly break down, or an acid breakdown, which facilitates the conversion of the polysaccharides or the complex carbohydrates into simpler sugars. And then the final step, of course, is the fermentation step, and that's the one that is the focus of this particular video. This really is the process, and it's important that you remember this equation, and then you can reproduce it when you're asked. So this is glucose, C6H12O6. It's not the only compound with this formula, but for our purposes, glucose is fine. Yeast, and particularly the enzymes in yeast, are what are involved in converting this. You can see there's no oxygen on this side, no oxygen required. And what we get is for each 1 mole of glucose that is fermented in this way, we get 2 moles of ethanol, and we also get 2 moles of carbon dioxide. And this is quite useful because this gas will manifest itself as bubbles or bubbling. And so we can see that our reaction is proceeding just by bubbles being produced in aeration mixture. And you'll do this in just a conical flask, something like that. Fermentation can produce solutions up to about 15% alcohol by volume. Beyond that, it kills the yeast. And so therefore they can't ferment, we can't produce anything with a higher alcohol content than 15%. Of course, if we're going to use this as a fuel source, the fuel must be much higher than that. So if we want to obtain something like 95% ethanol, then we have to use the process of distillation in order to do that. Because ethanol has a boiling point of roughly 78 degrees, it boils at a lower temperature than water, and therefore it can be separated first. Just the different fractions can be separated by the fact that ethanol has a boiling point of 78 and water 100. So therefore, as long as we control the heat, we can get our ethanol out and leave the water behind. The main reason that we're looking at this is because not only is ethanol such an important compound, but the process of fermentation allows us to use it not only for a fuel source, but also in the manufacture of polymers. And here's a little sort of diagram to give you a bit of an overview of how this works. So we start with a source of starch or cellulose that can be corn or sugarcane. We use enzymes in order to break that down to produce the simpler sugars, which is the glucose. So these are complex and these are simple. Through fermentation process, that's what gives us ethanol. We can dehydrate the ethanol to give us ethylene or ethene, and this has been our key starting project compound for most of the products that we've been looking at in this topic. Through substitution reaction, for example, we may produce chloroethene and then ultimately the polymer PVC. And this is just one way of tracking how we can go from biological materials to our polymers. Thanks for watching.